Each of the cell organelles one at a time, and we'll make a few points about them. And you know, if you can scribble down at least a couple of the important ones, then I think you got a pretty good comprehensive summary of what we need to know about these different cell organelles. Definitely at this stage, right? We'll be coming back to them, of course, again and again. But we already said, we talked about last week, we talked about lipid structure, triglycerides. So these are modified triglycerides that are what we call phospholipids. So they have, instead of 3 fatty acid chains, they have 2. And one of the fatty acid chains is kind of replaced by a phosphate, which is attached to the glycerol. So we call these glycerol phosphate heads, right? And you can see on the right-hand side, I've circled off one of what we call a phospholipid. So these are not technically triglycerides, right? These are modified so that there are now only 2 fatty acid chains and a glycerol phosphate head. We call that a phospholipid. But you notice the way they're arranged. They're arranged side by side. These each phospholipid is right next door, shoulder to shoulder, beside another phospholipid, and the tails, what we call the tails, are dangling down. But then, and they form this you know, this row, if you like, of phospholipids with all the tails facing downwards. But they're met or matched by another pair of fatty acid tails from another layer of phospholipids, and this forms what we call the phospholipid bilayer. So cell membranes are composed of a phospholipid bilayer in which there are embedded proteins. Sometimes those proteins literally cross the whole of the double membrane. Sometimes they're just sitting on the outside or sitting on the inside, sort of stuck into the membrane, but sometimes they form these channels, what we call protein pores. And these are partly what gives the function, what gives the cell membrane its function. So the chief function of the cell membrane is that it regulates or controls what substances can get in and out of the cell. So we're talking about all these kind of molecules. A lot of them are molecules that you eat. So, you know, all of the finely digested biomolecules, the subunits that end up going into the bloodstream and on off to the cells, they're taken into the cell, usually by diffusion, sometimes by active transport. In other words, they need to be actively taken into the cell. And that's where these proteins, these selectively permeable proteins, come into play. So they are channels through which certain molecules can pass. And sometimes it costs energy to get those in. That's what we call active transport. ATP is needed to get those things into the cell because it's against a concentration gradient. We're doing movement through membranes next week, right? So either by diffusion or active transport, materials get in and out of the cell. And that's what the job of this is. One thing to notice is it's not like a container really, right? It's not like a plastic bag. It's not like cling film. It's more like in sort of, you know, physically, what would it appear like if we were to be able to be that small and we could look at it? Actually, it's a flowing kind of, it's a fluid thing. It's more like if you drop if you know what it's like to drop oil onto water. If you drop oil into water, it kind of spreads out sitting on top of the water, forms a really thin layer. That's more like what the cell membrane is. It's a fluid bilayer. Okay, a fluid phospholipid bilayer which contains proteins. It's not as yeah. So that's what it is. It's more oily than rigid. Okay, have a quick look at this. Quick test, or just a quiz. What are the molecules labeled A and B? What do we call those? What's A? What's B? Now it's interesting. You're looking at B going, what is it actually pointing to? Give it your best shot, and I'll give you the marking scheme in a second. Molecules A and B. Very brilliant. Yeah, very good. Excellent, yeah. Okay, super. So it turns out that B was kind of, it's not very clear. What do they want? Do they want fatty acid? Because there's two fatty acid chains. That's one fatty acid chain that it's pointing to. Do they want lipid? Do they want phospholipid? So it turns out that what was accepted was fatty acid chain or phospholipid or lipid, which was very easy going off them. A was a protein. So B was the lipid, and A protein was enough. All you had to say was protein. Molecules, name the two such processes. They're the two processes I just mentioned. So if you can remember what they are, just stick them in the chat box there. What are the two processes, or I named two. You could actually, there's another one which they gave marks for as well. Two processes by which molecules can get in and out or through cell membranes. What are the two processes? We're going to be looking at two of them in detail next week, really, I suppose. Well, one in particular. Yeah, very good. Okay, so interesting. So here are the three answers that were accepted. Two of these three were accepted, and only these. And I don't know, it's interesting, somebody put passive transport. I don't think that was, as a process, it wasn't accepted. Right? That seems a bit mean. The process, what was accepted was diffusion, which is a process, and it's a type of passive transport for sure. Osmosis, which is actually just a special case of diffusion that only applies to water. So nothing can move by osmosis except water, because that's the definition of osmosis. It's the movement of water. But it's a case of diffusion, right? And the last one is active transport. So it's interesting. Somebody put passive transport in. I think that's a fine answer. Doesn't appear in the marking scheme. Was it accepted on the day? We don't know for sure. I'll see if I can find out. I don't remember because it's back a bit. But I'll have a look. The three answers on the marking scheme are passive transport. Diffusion, osmosis, active transport. Okay. The cytoplasm of the cytosol. This is a thing that seems to rear its head in the marking schemes. The SEC don't like, they want the liquid part of the cytoplasm to be referred to as the cytosol, because technically the cytoplasm is everything outside the nucleus, everything inside a cell except the nucleus and the vacuole. So the mitochondria is part of the cytoplasm, so are chloroplasts and so are ribosomes. They're all cytoplasm, right? They're all included in that. So cytosol, strictly speaking, is the fluid in which those things are bathed, right? That they bob around in. That's the cytosol. That's the fluid part. So I think we have to be careful about that. They sometimes are sticky about it. So what is that? Well, that's where most of the action happens. That's where most metabolic activities occur, is in the cytoplasm, right? Because it's everything outside the nucleus and the vacuole. So of course it is. It's where respiration happens in the mitochondria. It's where protein synthesis happens at the ribosomes in the cytoplasm. So, it's mostly water though. Oh, I'm going to skip this actually. Just carry on there. Okay. Plant vacuoles. Not much to say about plant vacuoles. They're storage organelles. In plants, they store mostly water, and they don't just, it's not just for storing, but actually a crucial role is played by the organelle, by the vacuoles, in support and shape. And the way this works is plant cells, water enters plant cells by osmosis, and it's we'll look at this more next week. Those vacuoles then swell up, a bit like a balloon inside a box. If you blow up a balloon inside a box, then the balloon is going to inflate, and it's going to start pushing out against the walls of the box, right? That's exactly what happens with the vacuole. So it swells with water, pressure builds up inside the vacuole, it presses against the cell membrane, which presses against the cell wall, and so the whole cell becomes what we call turgid. We'll look at that again in a bit more detail. So a role in support and shape, not structure, right? If they ask you what's the role of the plant vacuole or the role of the cell wall, you can't use the word structure as one of its functions. It's not a word that we apply to function. It's the word we apply to how things are put together, composed. So don't use the word structure. It's a common one, and they see it every single year. It's this thing that's seen on the exams, and they give out about it. So shape and support. Okay, that's the one of the roles for the plant vacuole. Similarly with the cell wall, right? It has a role in shape and support. You can't say it gives structure to the cell. That's not going to get you there. It gives shape and support to the cell. It's fine. What's it made of? It's made of cellulose. So that's its structure, right? It is, if you were talking about plant cell wall structure, its structure is it's made of cellulose. It's rigid. It's slightly elastic. Sorry, it's not rigid. It's tough, but it's slightly elastic. So it's got a bit of give in it because it's got to be able to, have this elasticity because it's going to be, under pressure from the turgidity of the cell. So in between the two plant cell walls, we talked about this a bit next week, and we mentioned it last week. There's this kind of cement that holds neighboring cells together. In plant structures and plant tissue, the cells are stuck together, and it's called the middle lamella is what the structure is there. Chloroplasts are these green ovoid membrane-bound structures. So in other words, they're surrounded by membrane. That membrane is a double membrane. Okay, so there's a double membrane around the chloroplast, as well as these stacked inner membranes, which we'll look at in a second. So its function, look at the structure more in a second, but the function is it contains chlorophyll to trap light. That's one of its functions. It manufactures glucose through the process of photosynthesis. That's the other major function. That's, if you're thinking about chloroplasts, should be straight away, it's side of photosynthesis. But don't forget also, they trap light. They trap light, and then they carry out this chemical reaction using the light energy to carry out photosynthesis. I mean, obviously one is part of the other, right? But it's good to kind of tease those two related and overlapping functions together. So the manufacturing glucose, that's one thing, that's a large part of what photosynthesis is. But the trapping chlorophyll, and if you've done photosynthesis in detail, you know that that's the light phase and the dark phase of photosynthesis. Light stage and the dark stage, I should say. So you only find chloroplasts in plants. I should have said that. There are other photosynthetic organisms. There are some bacteria, for example, that like cyanobacteria, they can photosynthesize. They don't have chloroplasts, though. You only find chloroplasts in plants. This is the kind of drawing you want to be drawing if you're asked to draw a diagram in an exam. Okay? You should always try and just stick to two-dimensional diagrams. And we'll do some drawing now in a week or two. We'll get into drawing in a big way. But for today, there's a double outer membrane, so two layers on the outside. Then those, and they don't even look like piles of coins. They don't look like stacked disc-shaped membranes in the inside there, right? But that's fine because this is your diagram. This is your two-dimensional diagram. You don't have to indicate that they're stacked piles of coins. That's what they look like in a transverse section, right? So you've got those stacked membranes. The stacks are called grana. One stack is called granum, called a granum, right? Like pile of coins is what they usually talk about. And each of those membranes, those disc-shaped membranes, is called a thylakoid. That's not a word we need to know, by the way. It's probably a word that you've heard maybe, but just to make sure that you don't confuse the three different things. So there's thylakoids, they're the disc-shaped membranes. Each one is a thylakoid. A stack of them is called a granum. And then you have loads of grana, plural, in a cell, right, in a plant cell. Sorry, in a chloroplast even. This is the 3D view. And this is not something you'd ever attempt to draw. It's something you could be asked to label, though. So the grana is the pile. You see the double membrane there, the double outer membrane. It's got an outer and inner membrane. And then you've got these stacked membranes on the inside as well as the double outer membrane. Okay. This is what I mean by this is a transverse section through a chloroplast. It's artificially colored, right? Taken with an electron microscope. So you can see the grana there, and you can see, you can actually see the double outer membrane if you look closely as well. So they like throwing electron micrographs in the exams every now and again, asking you to label them. So you should know how to, relate these to the kind of cartoon diagrams that we tend to generally study. Mitochondria then, moving on to mitochondria. Mitochondria is the site of respiration. That's the function of the mitochondria. So what happens there? Respiration is the release of energy from food. So glucose generally, but not just glucose, other sugars, and also even things like fats, or fatty acids rather, things like glycerol, things like amino acids can be respired here, can be broken into smaller molecules broken down in catabolic respiration to release the energy that's contained within the bonds that hold them together, right? So they're all high energy molecules. They can all be respired. What's produced there is ATP. So they're the site of respiration. That's what one of their functions is. They produce ATP. It's really important to say that. That's the point of respiration, right? Is to release the energy in glucose or similar molecules as ATP, which can then be used in, cellular activities. So there's kind of two related points as well. Then as regards to function, the inner membrane, again, it's got a double outer membrane. Sorry, it has an outer membrane, and then the inner membrane is folded. So it's a double membrane, but it's different in the way that's laid out in chloroplast, right? There's an outer membrane, and then there's an inner folded membrane. Now the foldings are to increase surface area, and it's actually that's the same kind of thing that's evolved in chloroplasts. Those stacks of coins and the disc shapes and all that, that's again sort of increases surface area rather than just having a single membrane. These foldings add lots of surface area because on the surface of those membranes is where you'll find the electron transport molecules. Okay, on the surface of I should have said, on this wrong way. On the surface of these pile of coins, these grana here, on the surface of the thyroid membranes, the dish, that's where you'll find chlorophyll molecules. They sit on the outside like little trees, weirdly, waiting to be hit by photons of light so that they can absorb them, right? So that's where the grana are. That's where the chlorophyll molecules are in there. When you're looking at mitochondria, it's what's on those membranes is the electron transport molecules. Electron transport molecules are also on the membranes in chloroplasts. Different kind of electron transport molecules, but similar at the same time. And inside you've got this fluid called the matrix. The fluid matrix that's contained within the inner membrane. Okay. Both mitochondria and chloroplasts have their own DNA, and it's what we call extra chromosomal DNA. Outside the chromosome, there is some DNA. It's not all the DNA of the cell that's held in the nucleus. Some of it, is contained within mitochondria. What's on that DNA? Basically, genes that code for the enzymes or the other proteins that are needed for respiration, of which there's quite a few. And similarly with photosynthesis, the dark phase needs enzymes to happen, and the genes for those are contained within the small circular DNA that's within the chloroplast. So here's an EM of mitochondrion. Similarly, it featured in an exam one year. Why can you not see that image using a light microscope? What's the answer they wanted there, do you think? What do you think? Why is it that you can't see that image? I mean, we kind of know, but what's the answer they're looking for? You're wondering. Yeah, good. A lot of you are kind of giving, sophisticated answers saying, well, the magnification of a light microscope is not sufficient to see mitochondria. Good point. They actually let you away with just going mitochondria are too small in the end. Often what happens with market schemes is they were looking for something bigger, and then the answers were so disappointing that they just said, oh, okay, fine. We'll accept mitochondria are smaller or too small. That was fine. Name one type of animal cell that does not have mitochondria. Tricky one. So which cells that you can think of, an animal cell that doesn't have mitochondria, and why would that be? Why would there be cells that don't have animal cells that don't have mitochondria? Why is that about? What's that about? Okay, good. If we can just put our answers in the direct message rather than everyone, and then people have a chance to have a think about it before the answer appears. So the answer is, for one example, is red blood cells. Red blood cells don't contain mitochondria, and there's a very good reason for that. Is because their job is to carry oxygen around the place, and if they were carrying oxygen around the place, that oxygen would be used up by the mitochondria. So, makes sense for them not to have mitochondria then, because then they're using up the very stuff that they're supposed to be transporting. So the other thing is they don't really need mitochondria because they don't live very long. right? It's an unfortunate life being a red blood cell. You don't get to live very long, maybe 100 days. Then you're just broken down and recycled in the liver, so in the spleen and the liver. So you don't need mitochondria, right? You don't qualify if you're a red blood cell. Poor red blood cells. Nucleus, our last bit. What goes on in the nucleus? So again, structure, nuclear membrane, double membrane, bilayer. It's just exactly the same kind of setup as the other membranes, right? All the other membranes we talked about are just lipid bilayers. They're phospholipid bilayers, just like the cell membrane. They're actually not really structurally that different. This does have protein pores in it, which are they're structurally a bit different to other pores that you might see in other membranes. So it has nuclear pores in it. We call this double membrane, or the outer membrane we call it the nuclear envelope, but it's just a membrane. It contains chromosomes, or chromatin in the loose form, which is made of DNA and protein, held together with protein. Okay, but it's mostly about the DNA. The protein is just packaging. Okay, the DNA contains genes. What genes are stretches or sections or regions of DNA on the chromosome, parts of the DNA that encode proteins. Not all the DNA, in fact, most of it, not an awful lot of it, doesn't actually, encode protein. So it's not genes technically. And the bits in between the genes we call non-coding regions. So those bits that are genes we call coding regions because they code for protein. And genes, then the definition is it's a section or a region or a section of DNA that codes for a protein, codes for the synthesis of a protein or the manufacture of a protein, something along those lines. Where are those proteins made? They're made at the ribosome. But the chromosomes can't get out through those nuclear pores. All that can get out are small, temporary copies, transcripts of the genes. And we call those messenger RNA molecules. We'll come to that when we do protein synthesis later on. But for now, just know that the information is contained in here. All the information for protein making is contained within the genes. So how does it get out to where the ribosomes are? Because they're in the cytoplasm. So the answer is they get out as small, temporary, single-stranded transcripts, copies of the genes called messenger RNA molecules that diffuse out through the nuclear pores and then interact with the ribosomes. Ribosome, I'm just going to skip this because we're doing it next week. Ribosomes are where you get proteins being made. It's the site of protein synthesis. They're composed of about a 50-50 mix of RNA, ribonucleic acid, and protein. But here's the interesting thing: it's the RNA that does the catalyzing. It's the enzyme. Weirdly, we'll see when we do enzymes that we thought that all enzymes are proteins. Well, here's an exception: the RNA in the ribosome is actually the enzyme that's doing the work. They're visible just as small little grains in the